Okay, good afternoon. I did post yesterday some practice questions for the exam. So make sure you take a look at those. There's some really good questions there that I thought, man, I should have saved this for the test. But there you go. Take a look at those. They're on Blackboard under the tab Learning Objectives and Practice Exam Questions. Included the answers as well. Speaking of objectives, the vaccine, so we're going to finish up vaccines today, just a little bit more to finish there. We did go through how the COVID vaccines work, and we're going to talk about the malaria vaccine. So I didn't mention those in an objective, but because I didn't really anticipate talking about that in the summer. So just make sure you review that material if you pay close attention to those objectives. So exam on Tuesday. Innate adaptive immunity, so chapter 15, 16, the little bit of chapter 17, the vaccines, and then today we're going to talk about staphylococcus. Okay, so that's what will be included on Tuesday. I said that in an announcement as well. The homework for staff won't be due until the final. So you can go in and look at some of those staff questions. They're mixed in with other questions about streptococcus and bacillus and some other stuff. So that's why I'll just make it all due later instead of kind of making a separate assignment there. So that just seemed easier. Are there questions from last time? That was probably the most difficult section, I think, in the whole semester when you take what we learned about MHC class 1 and MHC class 2 antigen presentation and put that together with how T cells and B cells are activated. All right, so we do have another question, yes?
Question 8, the gluten type A,
okay, so one of the practice questions, that was an exam question last year. It was, I told you about it's actually a disease called gluten agammaglobulinemia. I can't hardly say it. So I threw that at you. It's a kind of a application type question. Say somebody has a disease where they don't make IgA antibodies. How would that affect them? And I think basically, if you know that IgA antibodies are the ones that are most prevalent on our mucous membranes, okay, then you're not going to have this good defense against infections that are that's going to be their portal of entry. So I think the question was not, it was a little bit more specific than that, but that's the gist of it there. Okay, I will be in my office all morning tomorrow and early afternoon. So if you want to come in and chat about anything, adaptive immunity, anything else. If you have told me that you want to take a makeup, want to take the exam before Tuesday and have not settled on a time, make sure you connect with me about that so I can try to reserve a quiet place for you to take that. Okay. All right, so when we ended last time, we were talking about how do vaccines get approved, all right? In this country, it's the FDA, the Food and Drug Administration, that does the, looks at all the data and decides whether a vaccine should be approved or not. Does it work? Does it do what we want it to do? And is it safe? That's primarily what they're looking at. So you might have heard about, clinical trials. That was the big hubbub. Oh, we rushed this through. Are they really safe? All those, there were over 100 different vaccines that were being developed, and we had to figure out, what are the ones that work the best. and what are the ones that are safest. We went through three clinical trials, and before that we did some testing on animals and just made sure that the vaccine itself wasn't harmful to the animal. And they look at, you know, in increasingly larger groups, phase 1, small group, healthy people, phase 2, a lot more people, phase 3, thousands of people. They look at, did the vaccine work? What kind of side effects were there? We changed the dose around, try to get the dose just right. And after phase 3, there has to be a period of 2 months where you're looking to see were there any adverse effects. So yes, this was rushed a little bit, only in the sense that for the COVID vaccines, we kind of started phase 2 before we had all the data in from phase 1. And we started phase 3, maybe a little bit before we had kind of accumulated all the data from phase 2. We still did all 3 of those phases. Okay, so at that point, if the FDA decides that it's safe and that it works, then FDA will approve. But after that, there's another phase that's ongoing. Okay, so phase 4, we call it the VARS system. So that stands for Vaccine Adverse Effects Reporting System. So you may have had a chance to opt into that when you had your COVID vaccine. I know I got a text about it. Basically, if you opted into that reporting system, you got texts, you know, in the days following your vaccine to ask if you'd had any side effects and how severe those side effects were. Okay, so just to be another data point to try to understand, you know, how safe these vaccines are. So basically, you know, in the past, before we were so connected with cell phones, if you went to the doctor because of some adverse effect, then that would get reported. All right, and that's ongoing, and that's how, you know, we can understand, again, hopefully understand how safe these vaccines are. Typically, any side effects are within the first 2 months, usually within the first couple days, the inflammation. Okay. One more vaccine that I want to talk about, and this news just broke on this about a month ago, but apparently we've been working on these vaccines for a long time. And I wasn't aware that we were as close to, you know, having a good malaria vaccine. So the malaria vaccine, and I forgot to write this on here for malaria. It's considered a recombinant vaccine. And that just means that we're using molecular biology techniques to kind of fuse some molecules together. We had to use some recombinant techniques with the RNA and the DNA viruses, but we just kind of call those a little bit, I don't know, we name them a little bit different. Okay, so a recombinant vaccine, it's called RTSS, and then the trade name is Mosquirix, mosquito, and so the trade name is a little bit more intuitive why we call it that. This seems like an odd name, RTSS. It made sense to the people that developed it. Let me get back to why they called it that in a minute. Okay, so they fused several proteins together to try to get our immune system to notice those proteins and then make memory cells, so that when it's encountered again in an individual, it will have a good adaptive response right away. So they remember the causative agent of the most severe kind of malaria is Plasmodium falciparum. Okay, that's the kind that we see in sub-Saharan Africa. And they've taken a protein. It's a protein within that protozoa called circumsporocyte protein. And if you remember a little bit, this might be a little stretch because we didn't emphasize this, but the life cycle of malaria, once a patient is bit with a mosquito that's carrying that protozoa, it will have the first stage of its life cycle in us in the liver, right? So it attacks hepatocytes in the liver. And then once it matures, we have all these different names for the different kind of stages of the life cycle of the protozoa. Once it matures, then it enters our red blood cells. And that's when we start seeing the effects of the disease, like the high fevers and the cyclic nature. So this is a protein that actually is necessary for motility and entry into liver cells. Okay, so we're attacking this protein that's necessary even before they get into that red blood cell stage where it's causing us damage. And they actually found that I think this is a protein that's within the protozoa. So it's not something that our immune system necessarily would recognize very easy. So they had to add something to that so our immune cells would recognize it. So they added a surface antigen from hepatitis B virus. I don't know how they decided to use something from hepatitis B, but again, it was right on the surface, so it had good antigenicity. We had to add that to get our immune system to notice it. So this is where the name comes from. The R, and I would never ask you this, but in case you're curious about it, the R stands for repeating, let me get this right, the genes from the repeat of this protein on the protozoa, and the T stands for the epitopes that our T cells recognize from that protein when they're presented, MHC class 1 presented on the surface. And then the S stands for these surface antigens. So they've squished those three proteins, I guess, together or recombinantly and made those into one molecule. And then they've added more of that surface antigen. So RTS altogether plus more of that S. That's where the name comes from. The whole idea is to get antibodies titers high enough to that protein to keep the parasite from ever infecting the liver. All right, and how good does it work? Initially, we said, oh, this works about 30% of the time. Another source said somewhere between 30 and 50% of the time. So it doesn't seem like it's that great of a vaccine, but when you consider in sub-Saharan Africa, there's about 250,000 deaths of children a year due to malaria, that if we could cut down on that by even 30%, say 3 out of 10 of those kids, then that's going to be something that's really quite exciting. So downside, 4 injections. So that's kind of a challenge in rural areas trying to get kids, 4 different, 1 injection and 3 boosters. So that's again really a challenge, but they've been testing this for quite a while. So since 2019, over 800,000 kids in Kenya and Malawi. So and there's more vaccines in the work, one that they even think is going to work even better than this. So that's pretty exciting. All right, so just to wrap up our vaccines. So there's always going to be some controversy about vaccines. There's always going to be people that value their own autonomy more than responsibility to other people. But what you can't argue with is the numbers in terms of 2 infections, polio and measles. So before we had the polio vaccine, we were around 40,000 cases in the US. It doesn't sound like a lot, but that's, severe, crippling, sometimes deadly disease in children. After, the polio vaccine came out, we had a couple different kinds. We talked about that. We haven't actually had any polio in the US for 30 years. We haven't eradicated it yet worldwide. It's still in Afghanistan, Pakistan, and Nigeria. And even there in those countries, the numbers are coming down. So we're hoping to totally eradicate that disease, just like we did smallpox. Measles vaccine, over Thanksgiving, ask your parents or grandparents, probably not your parents, ask your grandparents if they had measles as a child. Because before the vaccines came out, before, in 1960s, whatever, almost everybody had measles because it was so contagious. Complications were rare in this country with kind of good, medical care, but they were possible. And, incidence now, like way down, way down. So we just can't argue that, vaccines have saved a lot of lives. Okay, so I won't preach at you anymore about vaccines, but I'd be glad to have conversations. Any of you have questions, certainly, or comments about that. All right. So unless there's any other questions, we'll move on. We're going to start a new unit now, what we like to call our bug parade. Okay, so at the end of the semester, we basically, whatever time we have left, we talk about interesting pathogens and look at them in a little bit more detail. So I will, during Thanksgiving, I'm going to work on kind of deciding which ones that we'll talk about this year, which ones we have time for. So I'll get you some information after Thanksgiving. Hey, these are the pathogens I anticipate talking about. You are only responsible for those. So you kind of, pick and choose. Don't read all of chapter. I mean, you can read all of chapter 19, but, look especially at the organisms that we're going to cover. And also talk a little bit more about what the final will look like. So we'll get some information about that coming soon. All right, so medically important gram-positive bacteria. There's 3 genera of cocci, especially, that we think about when we're talking about gram-positives. We're going to talk a little in depth about the staph and the strep. And so if you're in lab, you're probably thinking, yay, I'm already familiar with a little bit of this. So there is quite a bit of overlap there. The enterococcus used to be considered streptococcus, and because the streptococcus genera was so big, they kind of teased that off about 30 or 40 years ago. We're not going to talk anymore about enterococcus, other than, hey, they're pretty much share a lot of same things with streptococcus. All right, so what can we say in general about staphylococcus characteristics? They grow in grape-like clusters. So what I like about this unit and talking about the pathogens is that we get to kind of go back and review some stuff that we've talked about earlier in the semester. So remember, and I'll go back to that other slide in just a second. Remember our cell arrangement patterns. Staph tend to grow in these grape-like clusters. Streptococcus, which we may have time to get to today, tends to grow in chains. So if you did a gram stain in lab this week and saw a lot of long chains, then you probably have streptococcus for your unknown. Now, if you don't see chains, don't automatically assume that you have staph. Some species of strep tend to chain up better than others. But again, you see those long chains, and that's pretty indicative of some kind of streptococcus. Okay. Where do we find staph? They're commonly on the skin, mucous membranes, or anything that you would touch. So light switches and keyboards and cell phones, you're going to find staph on there for sure. They are catalase positive. That's an enzyme that we have mentioned when we were talking about toxic oxygen, forms of toxic oxygen. So that hydrogen peroxide can be formed as a result of aerobic respiration. Oftentimes it's the superoxide that gets detoxified into hydrogen peroxide, which in hydrogen peroxide still, again, is toxic to cells. So cells have to have a way to detoxify it. Where does it come from? Well, sometimes the electrons are kind of leaking back into the membrane where they shouldn't be, or oxygen doesn't split quite right during that last stage of aerobic respiration. All right, so again, catalase breaks down that hydrogen peroxide into water and oxygen. It's a really nice, easy test that we do. To test for catalase, we put our organism on the slide and then add a drop of hydrogen peroxide, and we look for that oxygen that comes off. Vigorous bubbling is a positive. All right, if you're in lab, you know that. Let's see, what else? Some of them form slime layers. or perhaps even a capsule. All right, so that's just, so a lot of organisms do that. It's just tremendously helpful for them to, when they are inside a host, especially. Let's see, what else? They are facultative anaerobes. So they're pretty flexible. They can live in the presence of oxygen. They can metabolize without it, either by fermentation or anaerobic respiration. And they are somewhat environmentally resistant. Now, they don't make spores, and they don't, there's nothing special about their cell wall, like they're not acid fast or anything like that. But when you compare these organisms to other, vegetative cells, like you compare staph to strep, the strep are really wimpy in the environment. Where the staph can do things like they are fairly salt tolerant. They are going to be able to live in places with like pH fluctuation, temperature fluctuations. Okay. So they're just, they're not super sensitive to all that stuff. They just like, oh yeah, whatever, I can live in that. Okay. We're going to zero in right away here on one species in particular, and that's Staph aureus. We'll mention a couple other ones at the end, but Staph aureus is really the one that's clinically the most interesting. There are over 30 different species of staph. There's a lot out there, but then this is the one that kind of you always hear about as always causing infections. Okay, so where do we find this organism? A little bit more about it. It is actually pretty common for it to be on our skin, upper respiratory tract, nose, mouth, places like that. Sometimes we consider it an opportunistic pathogen. Sometimes we would, scientists don't agree, basically. Sometimes it's called an opportunist, and sometimes it's considered a true pathogen. The reason that we might call it an opportunist is because it typically takes a very high infectious dose. So you just get a few staph in a wound, Staph aureus, right? It's not going to cause anything. You need, we think, at least 1 million cells in there. So it's pretty easy to get 1 million cells, if you're ingesting some food or contaminated water or milk or something like that. But again, because the infectious dose is high, sometimes it's considered an opportunist. So where might we get a Staph aureus infection? Commonly transmitted in hospitals. So things like catheters or implants. Okay. And when it's spread by close contact in the community, that's typically going to be, maybe some kind of skin infection. So, a breakout, on the wrestling squad or in a locker room or something like that. Okay. So let's see. And how is it spread? I should talk about that. Usually by large droplets. Okay, so this is not something that's airborne. Typically you're in close contact with somebody else, within a meter of them. Those large droplets that we would, sneeze out or cough out, most likely. Not going to be airborne, not typically transferred by animals. Like there's not typically animal reservoirs that I'm aware of. That may be, not unheard of, but it's not the most common way that we would get a staph infection. Usually animals have their own staph and then they don't pass their kind on to us. All right, so usually when we get to talking about the actual infections, usually those are some things that you've heard of before. Probably the virulence factors that make the pathogen successful is the thing that students have to kind of study a little bit more. But the nice thing is, again, we kind of draw in things that we've talked about already. So it's a nice review of the semester. So remember, what virulence factors are. They're what makes the organism successful in establishing an infection. A couple structural virulence factors. Again, we're talking about staph aureus in particular here. It has, whether you call it a capsule or a slime layer, same thing, some kind of a layer around it that helps inhibit definitely phagocytosis, especially by neutrophils for some reason. I'm not really sure. This I copied this right from the book. Why it would inhibit chemotaxis, unless maybe our white blood cells might kind of get caught and stuck to the capsule of the Staph aureus and keep it from migrating somewhere. I'm not really sure. But we do know that it will increase the chances that organism can colonize some kind of implant or catheter. We hear about that with MRSA quite a bit. I'll talk about that in a minute, but that's a Staph aureus infection that after surgery, sometimes you just can't get rid of it. You have to take that implant out and start over again. So besides a capsule, another virulence factor that Staph aureus has is protein A. All right, so this is something that's on, again, the surface of that Staph aureus cell. All right, so I've got the picture here. It's protein A is this little, is signified by these little orange dots here. And that's picture it all over the surface of the cell. And what protein A does, that it binds to the Fc region of antibody. So remember your parts of the antibody, the stem region here, that's the Fc region. And remember, this up here is the fragment of antibody binding. Heavy chain, light chain, remember. All right, so if protein A sticks to that Fc region, you can see how we've got the antibody held like this. Okay, so what's the significance of that? Remember, the business end of the antibody is this part up here. This is a part that binds to epitopes. And then it may bind to two different epitopes of one cell, one of another, and start to clump those cells together or agglutinate them. And then they'll get filtered out in the lymph or in the spleen. Or it might, the antibodies might bind and neutralize some kind of surface receptor. When the antibody is bound like this with the Fc region, then it's kind of, it can't do anything because it's not binding to the antigen. So it's a kind of a good strategy to keep antibodies from doing what they need to do. Okay. Kind of like if you were going to catch a snake, okay, you come up behind it and grab it by its head so you don't get the fangs, right? So same idea, or that works in my brain anyway. How about some exoenzymes? So some of these, again, should be familiar. Remember, these are virulence factors that are secreted outside of the cell and stay pretty close to that Staph aureus cell. They don't really go systemic in any way. Coagulase, another one that you're familiar with if you are in lab. What coagulase does is clots blood. All right, and that's an advantage to the organism. Say it's infecting a wound and it's going to try to kind of hide from our immune system. It makes a blood clot around it, kind of keep our neutrophils and macrophages from those first responders from getting to that site of infection. So clots the blood, kind of walls itself off, and again, helps the organism establish itself in the host. Okay, and we test for coagulase. Again, just kind of a reminder, those of you in lab, we inoculate rabbit plasma and incubate it. And then if we see a clot in that plasma, which is, remember, the liquid part of blood that would have all the clotting factors in it, if we see a clot here, that's going to be a positive coagulase test. Staphylokinase, this is then how the Staph aureus would break out of its clot. So it's made a clot, it's reproducing, getting a lot more of it. And when it uses up all the nutrients there, it's going to need to break out so it can, invade further into the tissues. So this is going to dissolve the blood clots. Hyaluronidase is one that we talked about before. This is a exoenzyme that's going to break down a matrix between cells. We talked about hyaluronic acid as being kind of the glue that holds our cells together. All right, so again, thinking invasion, breakdown, invade deeper into our cells. Lipase, another thing you tested for in lab, is going to hydrolyze lipids. Those bonds in lipids. And why would that be useful? Our skin can be kind of an oily place, right? We have sebum on our skin, and organisms that want to live on our skin, it'd be helpful to be able to break through those oils. Okay, so we're going to talk about one particular infection where that lipase is important to, again, invade deeper. Some of our staphs are able to make some kind of beta-lactamase. And remember, that's important in terms of being giving it resistance to beta-lactam antibiotics. Okay, so no one strain of Staph aureus is going to have all these things. Different strains, might have the ability to make different of these enzymes. Okay. And then there's some exotoxins as well. I'm not going to make you write quite as much on this page. Remember, it doesn't have endotoxin. It's only the gram negatives that have endotoxin, but they just definitely secrete some exotoxins. Staph aureus actually does produce hemolysins. And if you're in lab, you should be thinking, I thought that was a streptococcus thing. Well, it turns out staph can lyse red blood cells. So when you streak out Staph aureus on blood agar, it's actually beta hemolytic. It totally lyses red blood cells. You can see this yellowish here, breaking down those red blood cells. Okay, it's more of a in vitro observation. So we see that when we're growing them in a petri dish in the lab. We don't think that has a lot of effect in our bodies. Like it sounds like, ooh, lysing red blood cells, that sounds bad, but it doesn't seem like it has a huge effect in our bodies. Some strains can make enterotoxin. Remember what that prefix means. Entero means intestinal. All right, so this is toxins that are gonna induce nausea and diarrhea by binding in our digestive tract. Okay, that toxin is heat stable. So if you have some food that's been contaminated with Staph aureus, and maybe then you heat it up and kill the Staph aureus, the toxins will still be there. So if the organism was growing up for long enough, just heating up that food is not going to be enough. Exfoliative toxin, that's a nasty one. We'll show the effects of that in a minute, but it causes our skin cells to separate and pull apart. And then toxic shock syndrome toxin. That is considered a superantigen. So remember, superantigens cause widespread T cell activation, kind of non-specifically. So maybe 20% of a person's T cells get activated rather than just 1 or 2 with a specific T cell receptor. So that causes massive cytokine production, uncontrolled inflammation. So that's a bad one. Again, it's only certain strains, and that's pretty rare, thankfully. So we'll talk more about that in a minute as well. All right, so looking at some common Staph aureus infections. So we'll start with the skin infections and then talk about a few others as well. Folliculitis, so that's infection of a hair follicle. Usually, so what does that look like? It tends to look a little bit pussy. So if you look, over here, the organism is using lipase again, to break down some of the oils in our skin and to help it invade, kind of sneak in around that hair follicle. So we get these kind of, little pussy bumps there. When are you likely to get folliculitis? Apparently, it's more likely on skin, like in a shaving area, or if you wear really tight clothes, right? So think about how tight those skinny jeans should be, I guess. So just skin that's been, abraded just a little bit, or you've got, a little cut in the skin or something as well. Or if you've got eczema or something like that, that's a kind of a risk factor. Okay, irritated, that was the word I was looking for. Skin that's been irritated. So like shaving areas or wearing tight clothes. Typically, this could clear up on its own, typically not something too serious. When it doesn't clear up on its own and it gets worse, then we could say it could look more like this. We would call this either a furuncle or a boil, okay, where you've got a spot that's looks really red and angry, kind of raised up, maybe a little pussy there. At this point, anytime you have a skin infection that doesn't appear to be healing on its own, after a certain number of days, it doesn't seem to be healing up, then it's appropriate to get treatment. Sometimes these boils need to be kind of drained by a physician. You might need some antibiotics to clear them up. Otherwise, they're going to scar. So, and if the furuncles, if these gets even worse, if they kind of get more than one of them all in the same place, we call them carbuncles. And this is getting serious. This is getting, the potential for this infection to maybe even get into bloodstream and go systemic. Common place for carbuncles apparently is the back of the neck. I don't know, maybe that's somewhere we just don't normally clean very well. I don't know, but that seems to be a common place. One other skin infection that is not serious, that typically doesn't ever go systemic or anything like that, it's a common childhood skin infection called impetigo. Basically, this looks like kind of open sores on the skin. Usually it's on the face, and those sores kind of scab over, and then the scab falls off and it doesn't heal up. It just kind of stays raw and angry. It spreads pretty easily. One kid maybe scratching his face and, touching other kids as well. So preschools, things like that. They really need to get treatment with antibiotics, again, in a timely manner so it doesn't scar. Okay, it takes a really long time to heal up on its own. My son has a scar right in the middle of his forehead here because I didn't realize that he had impetigo, and I just thought he was, scratching at his scabs and picking them off. So I can't see it very well anymore, but. All right, we have to mention MRSA. All right, methicillin-resistant staph aureus. This is a strain of staph aureus that's not necessarily more invasive or more dangerous than other strains of staph aureus, but it is very difficult to treat because it's got this beta-lactamase production. So it's resistant to most of the antibiotics that we commonly use. So all the beta-lactams plus others. Oftentimes that presents as a skin infection. So can you tell by looking at a skin infection whether it's MRSA or not? Not really. I picked this up off of WebMD. The difference between, sometimes you get a little something on your skin and you can't figure out where it came from. Oh, maybe it was a spider bite. Okay, so I don't know. Can you tell the difference? MRSA tends to be kind of pussy, right? Where a spider bite wouldn't be. Apparently spider bites are really not very common around here. So here's another example. That looks really quite angry. Definitely would want to have that checked out. Hopefully that lots of signs of inflammation there. There are two kinds of MRSA, the hospital acquired, the community acquired. Community acquired, again, is usually a skin infection. Hospital acquired can be more, things on catheters and implants and things like that tend to be more serious. The incidence of this, for some reason, is going down. We don't really know why, just sometimes, normally strains of something kind of replace themselves with other strains. It's still a very serious infection, but again, it's just a little bit more rare than what it was maybe 10 years ago, and we're not quite sure why. And then as we think about some more serious infections of Staph aureus, typically a patient is not going to experience any of these unless they're maybe got something else going on, like they're malnourished or they're immunocompromised or something like that. So again, if you're otherwise healthy, you're not likely to come down with any of these infections, but they can all result if the Staph aureus gets in your bloodstream. So if it gets in your bloodstream, you can get to anywhere in your body pretty much. You can have infection in the bone, osteomyelitis. And I didn't cite this picture very well. I do remember this picture on the left there was from a young Ethiopian girl. Had her femur was infected, so you can see, I don't think she could walk on that. Like, and for adults, it's more likely to have osteomyelitis in the vertebrae, just because of some particulars with the blood flow there. And I honestly cannot remember what this bone was. Maybe it's a arm bone. I'm not really sure. But what was I going to say about that? So these patients, they're going to need intravenous antibiotics for a long time. I'm not sure whether, if once it gets to this stage here, that's going to ever, fully heal. So it's pretty serious infections, very painful as well. Pneumonia, Staph aureus pneumonia is not common, but does have very high fatality rate. So even when we treat that with antibiotics, endocarditis, this is an infection of the heart valves, can cause a patient to have a heart valve replaced. Again, pretty serious. Meningitis, infection of the meninges, the covering of the spinal cord in the brain, bacterial meningitis, lots of things that cause that, or several different things anyway, very serious. And bacteremia, that's the same thing. That just means sepsis. So when you have an infection in the bloodstream, that's pretty serious because they can cause all these other things. So, okay. And lastly, some infections that we attribute specifically to toxins that Staph aureus produce. So again, one strain of Staph aureus wouldn't make all these different toxins. We see kind of some variation there. That exfoliative toxin can cause the scalded skin syndrome. So they put a little different picture in your book in the latest edition, but it's supposed to be very painful. It basically causes your skin to kind of come off in pieces. Obviously, that's going to leave you open to secondary infections because skin is our best barrier against a lot of pathogens. Apparently, this clears up pretty well if you're young on its own, but as you get older, we have a harder time clearing that toxin from the kidneys. So the kidneys start to shut down. So very serious infection. Food poisoning. Enterotoxin. Remember, entero meaning intestinal. That's one of the food poisonings that, pretty much right after you eat it, I think that wasn't good. I don't feel so good. Like, it can start to affect you within half an hour if you eat something. Sometimes the organism's got to grow up in your body until you really start feeling it, but that toxin will take effect pretty quickly after you eat it, and it's not serious. You're gonna feel lousy for 24 hours or so. And the real bad one here, toxic shock syndrome. So we said that was caused by the superantigen toxic shock syndrome toxin. What strains that produce that toxin? They are very rare now, thankfully. But in the 1980s, we had a big spike, 1980, of toxic shock syndrome. And we realized that was correlated with the use of a new tampon that came on the market called Rely. And those tampons were basically like a diaper. Like, the company advertised, oh, you can wear these for 10 hours and they won't leak. Yeah, but that provides a really nice, warm, blood-soaked environment for the Staph aureus to really proliferate. All that blood changes the pH in there to be conducive to Staph aureus growth. So that was a huge problem. Those were taken off the market, and now the incidence is really low. Men can get this as well as women. It's going to be as a result of maybe an infection during surgery or some kind of trauma. It's not usually not clear exactly where people get it. But what does it look like? Sudden onset of a really high fever. It's this uncontrolled inflammation. So, that the fever, other things like vomiting and diarrhea, a rash that's supposed to look like a sunburn. I can't really make that look like a sunburn, but it's not just a little speckly rash like you might see with some other infections. Even mental effects like confusion. Okay, so it causes the body to go into shock, meaning, right, the blood vessels get too leaky, too much inflammation, and your body's not getting enough oxygen. Okay. That's pretty much it for Staph aureus. The other two famous staphylococcus that I'll just mention here, they're the same ones that we used in lab. Staph epidermidis, pretty much not going to cause anything very interesting clinically. It's the normal microbiota on our skin, not really invasive. If it would get into the bloodstream, kind of the thing that it's most famous for causing is that endocarditis. But again, that's pretty rare. And Staph saprophyticus is best known for causing urinary tract infections. Apparently, for women of a certain age, it's fairly common. Why not as women get older than that? Apparently because they've usually already had it when they were younger, so they build up some immunity to it and aren't likely to get it as they age. Okay, so that's it for the staphs. So that's where we'll draw the line for exam 4. Okay. And sorry, I was kind of late getting streptococcus slides out to you. I was just not enough hours in the week this week for some reason. All right, so still in chapter 19, looking at the streptococcus. The streptococcus share a lot of characteristics with the staph. So sometimes, and it kind of sounds like staphylococcus, so it's a little bit easier to get those two mixed up. A lot of different species of strep, almost 50 of them. We're going to look at just a few of the most famous ones. But in general, these are gram-positive cocci. They typically arrange themselves in chains. Thus the picture here. Non-motile, just like the staph. In fact, I don't know of any round cells like strep, like any cocci that have flagella. There may be some exception to that, but for the most part, they do not. And they are non-spore forming as well, just like the staph. Most commonly facultative anaerobes. There are a few species that are truly anaerobic. We don't work with those in lab because it's too difficult. You can't have a petri dish of strep out in the air that's anaerobic and have them survive. Some of them are considered capnophiles. And that means that they grow best in a high concentration of carbon dioxide. All right, so when you put brackets around something, that means concentration, okay, elevated concentration of CO2. So when we're trying to grow streptococcus in the lab, we either put them in an incubator that's hooked up to a CO2 tank, or we put them in a candle jar, which is a much lower tech option, where we just take a jar and we put our plates in and light a candle in there. The candle, carbon dioxide is a product of combustion. All right, so that adds a little CO2 in there. The candle will go out when the oxygen gets used up. So we increase the CO2 concentration. Streps are catalase negative. So that's a big difference. Lots and lots of cells will produce catalase. A ton of them do, but streptococcus don't. So it's a nice way, if you know you have either staph or strep, to tell those organisms apart. They actually use a different enzyme that does the same thing. So this is what peroxidase does. It detoxifies hydrogen peroxide by using an electron carrier. All right, and then we end up with water and NAD. So there may be a way to test for that. There probably is, but it's a lot easier to test for catalase. Okay, so it's just another way to detoxify that hydrogen peroxide. All right, there's a couple of different ways that we like to categorize our streps. Again, if you are in lab, this should be pretty familiar. We like to streak them out on blood agar. Streps are a little bit fussy. They need a really rich medium to grow, like sheep's blood agar. And they have very characteristic ways that they look when they grow on sheep's blood agar. They'll have one of 3 different hemolysis patterns. Okay, in other words, characteristic ways that they lyse red blood cells. If they're alpha hemolytic, they turn that blood agar a little bit kind of brownish or greenish. So they partially lyse the red blood cells, but they don't lyse the hemoglobin. So the hemoglobin in the red blood cells gives it that little kind of greenish tinge. If our streps are beta hemolytic, they totally lyse the red blood cells and the light passes right through the medium. So it should look kind of yellowish when you hold it up to a light. And then the gamma is a little bit hard to see in this picture here, but streps that are gamma don't lyse the red blood cells at all. So they don't look, the media doesn't look any different at all around the cells. Okay, the other way that we use to tell our streps apart, or at least classify them, and don't memorize this, don't worry, is with Landsfield Groups. So Rebecca Landsfield in 1930s realized that Streptococcus had these carbohydrate surface antigens. They have sugars on their surface that can act as antigens. And that's unusual. We talked about how carbohydrates usually aren't very antigenic, but there's exceptions to everything. And so she realized that she could test this out using antibodies. So with her system, based on these antigens on the surface, most of our streps can be put in one group. So they go A down to S. For some reason, they skip some letters. I have no idea why. And in some of the groups, like Strep Pyogenes is the only organism that's in group A. But some of the groups, there's several different species in that group. And a couple of them, like here's an organism, this one is actually considered in 2 different groups. So it must have more than one kind of surface antigen. And to make this maybe even not a great system, we have some strep that don't have those surface antigens. So we call them non-Landsfield streptococci. So our strep mitis, our strep pneumonia, you've used those in the lab, they don't have those surface antigens. So I don't know, this system is in use, so I wanted you to kind of recognize it, but it does have its limitations. Sometimes we kind of group the hemolysis patterns taxonomy together with the Landsfield group. So sometimes Strep Pyogenes, we call it group A beta hemolytic strep. You see that sometimes GABHS or even GAS for group A streptococcus. So anyway, you see those abbreviations sometimes, and that's what they mean. So again, don't memorize that. Just kind of trying to explain what Landsfield groups are. Okay. I want to zero in on Strep Pyogenes primarily, and we'll talk about a couple other streps here today if we have time as well. So beta hemolytic group A, where do we find Strep Pyogenes? Mostly humans are passing it around. Maybe like 10% of us might have this as normal microbiota in our throat or on our skin. So not terribly common. It's going to be, again, transmitted by respiratory secretions. So those are the large droplets when you're within close proximity to somebody else. They're not airborne, not, again, usually passing back and forth between animals. Animals have streptococcus, but they're not the same ones that will infect us most of the time. There are some exceptions to that. I thought this slide was a little boring, so I included a picture of Strep Pyogenes. There's a website you can go to called giantmicrobes.com, and they sell plush microbes. So this guy is kind of cute. He has a fork and a knife on him, and we'll talk about why that is in just a minute. Kind of cute. if you're looking for that perfect gift, you know, coming up. Okay. All right, virulence factors. So again, we've mentioned a couple of these before. Starting structurally with the capsule. So, you know, benefits of a capsule keeps the organism from being phagocytized by our phagocytic cells, neutrophils and macrophages. What's special about the capsule of Strep Pyogenes, it's most bacterial capsules are made of polysaccharides. So if you think back to, if you just, again, for a second, if you're in lab, when we did a capsule stain, it's hard to get dye to stick to the capsule because it's made of these sugars that are not charged. So we stained the background instead for that one, if you remember. Specifically, Strep Pyogenes has a capsule that's made of hyaluronic acid, right? That's the carbohydrate, and that should sound familiar because that's, we talked about, this is the matrix that holds our cells together. So it's made of something that we find in our body as well. So our phagocytes usually don't notice it. They don't recognize that capsule as being something foreign. All right, so that helps it evade phagocytosis. Another anti-phagocytic factor that we talked about earlier as an example of an anti-phagocytic factor was M protein. And actually, I'm not sure what M stands for. It doesn't make sense that it would stand for membrane because there's a cell wall around the cell membrane here for these organisms. I think it might stand for motif protein. Anyway, Strep Pyogenes has the M protein all around on the outside of its cell. And there's a couple different variations of that we'll talk about. Primary function, it does a really good job of inhibiting the actions of complement. So especially, remember, there's 3 different activation pathways with complement. One activation pathway involves an antibody bound to an antigen that can bind to C1, and that will start the cascade. And one was the mannose-lectin pathway, where we got the sugars on the outside, mannose, of bacteria that's recognized by our proteins, lectin. The third way was that alternative pathway, where we said C3 is constantly being split a little bit, and then if it's in C3b is in the vicinity of a pathogen, it will bind to it. And along with some other factors that are usually there, can cause more C3 to split. Well, M protein actually inhibits activation by that alternative complement pathway. So as it's kind of splitting, the C3 doesn't bind because of the M protein to the cell, and it also inhibits some of these other proteins that have to be necessary for that activation pathway to begin. So we get less complement, no opsonization. Remember, the C3b will bind to the outside of the cell, and then our phagocytes can kind of grab a hold of that, and it's more likely to be phagocytized. So primarily that's going to help it resist being phagocytized by innate cells. And there's actually 2 different kinds of M protein. Like I said, it's more complicated than that, but there's rheumatogenic M protein. And that just means, we're going to talk about the effects of this later. Antibodies that we make to this M protein are going to cross-react, sometimes with heart antigens. Okay, so when we make antibodies to this rheumatogenic M protein, sometimes we get a little bit confused, and those actually target antibodies in our heart muscle as well as our heart valves. And then sometimes those antibodies actually cross-react with kidney cells. Oops. Or we'll say kidney antigens. Okay, so we'll talk about, we'll come back to that later, but you have a Strep Pyogenes infection that doesn't clear up very well, or we don't treat it very well, then there can be kind of autoimmune effects from that, right? So we'll pick that up later when we get to our infections. A few exoenzymes, I kind of picked and choose. I don't want to talk about all the ones that your book talks about. C5a peptidase. So that's an enzyme that Strep Pyogenes can secrete that does what you would guess. It neutralizes C5a. So remember, you know, that the complement can get activated in some other way besides that alternative pathway. C5a is a really potent chemotactic factor, as well as increasing inflammation in general. Okay, so it's going to, if you can neutralize C5a, that's going to decrease the number of white blood cells that migrate towards the infection. Streptokinase. So we talked about staphylokinase. This is the variety that Streptococcus has. Lyses blood cells. And actually, sometimes heart attack victims are given purified streptokinase if it's their first heart attack to try to break up a clot in the heart. It doesn't work on subsequent heart attacks because then our body makes antibodies to it, but we can actually use that for our good. The bacteria will use this to lyse clots. Maybe if you've got some tissue that's been damaged or injured in some way, then it helps it to kind of move through, you know, invade deeper into the tissues if there's some clots there. So it's just going to aid the spread of infection. And then another exoenzyme, sometimes you hear it called streptodornase, which is kind of a clunky term in my mind. We can just call it DNase. And why would that be good to break down DNA? Well, if you remember back to, this is a good review for the exam, the 3 ways that neutrophils can accomplish killing. They are phagocytic, they will degranulize, and they also can have this these what we call nets, neutrophil extracellular traps. And what they do is kind of break down their DNA and kind of expunge it from the neutrophils do from their cells to try to trap any invaders. Okay, so if the streptococcus can produce DNA to kind of digest that DNA, then they're not immobilized and they can kind of go on their way. So it's kind of frustrating, you know, you learn about all these tools that we have to fight pathogens, and then we learn, oh, then the pathogen has something to counteract that, you know. So it's kind of an arms race. So we do this and then they do that, and then and then a few toxins to mention as well. Streptolysin, so that's a hemolysin that specifically Streptococcus produce. We talked about that with the staph, that staph aureus can make hemolysins, but streps are probably even more famous for making hemolysins. And they act not just on red blood cells, but they actually can act on white blood cells and platelets as well. So causing kind of a lot of damage there. Why would they want to lyse those cells? Well, lots of nutrients from lysing a red blood cell in there, and they have this kind of dirty trick that when they invade a white blood cell, if they get phagocytized by a white blood cell, they can release streptolysins, which actually cause the lysosome to burst open before it fuses with the phagosome. So all those lysosomic enzymes get released within the cell, and sometimes that will kill that white blood cell, and then the streptococcus can go on its merry way. So kind of a dirty trick. Some strains, and luckily this is very rare now, of Strep Pyogenes, can make erythrogenic toxin. And this is considered a pyrogenic toxin. Pyro means fever inducing. Okay, but an erythrogenic toxin basically causes our blood capillaries to dilate a little bit near the surface. So it causes a red rash. It's actually a super antigen, another one, just like toxic shock syndrome, that's carried on a lysogenic phage. So remember, bacteriophage have two different ways, two different kind of life cycles that they use to reproduce themselves in a bacteria cell. They have the lytic cycle, where they just come in and then, you know, assemble themselves and burst out of the cell. And then they have that lysogenic cycle, where they will incorporate themselves into the host cell chromosome. Okay, so this toxin actually for Strep Pyogenes, if it's been infected with a bacteriophage, a lysogenic bacteriophage that carries this toxin with it, then the cell will actually, well, it causes a specific infection called scarlet fever, right? So only Strep Pyogenes that have been infected with this particular phage acquire the ability to make this toxin. So it changes their phenotype. Remember, we had that term lysogenic conversion, okay, where it actually changes their phenotype and enables them to do something that they couldn't do before. All right, so again, produces bright red rash all over the body. It's the kind of rash where you see it and you know you got to get to the doctor. It's like really kind of screams at you. High fever associated with that. So we'll talk about that in a second. All right, so keeping those virulence factors in mind, let's talk about some skin infections. Again, these are all possibly caused by Strep Pyogenes. Impetigo, we talked about this, can be caused by staph aureus as well. Impetigo caused by Strep Pyogenes looks very similar. Maybe some people can tell the difference that are used to looking at these things. It's supposed to be maybe a little bit more crusty lesions in nature, but still it's not serious. It tends to stay localized. I know it looks kind of bad there. Again, needs treatment with some antibiotic cream to get that to clear up before that's going to scar. A little bit more serious there, erysipelas. Not as common, but this is characterized, and it's usually children or elderly that get this kind of raised red lesions on the skin, oftentimes on the face, sometimes on the legs. It's called, those lesions are said to have kind of an orange peel consistency. So kind of the skin looks a little bit tougher and it gets these, you know, little tiny pits in it. Causes a really high fever. And usually a patient will get that maybe after they've had some mild trauma to the skin or a little bit of burn, but it only takes really, really tiny cracks in the skin for that organism to gain access. And it's pretty serious. Like sometimes this can be really mild and no big thing, or it could be life-threatening. So there's kind of a big range there. And a related condition to that is cellulitis. And sometimes it's pretty difficult to tell the difference between erysipelas and cellulitis. Cellulitis is supposed to be when the organism is invading a little bit deeper into the tissues. It's usually on the hands, face, legs. So you can see this example here, kind of the reddening here of the one hand compared to the other. Lots of inflammation going on there. Commonly enters through a break in the skin. Again, this can be life-threatening, so it's not something just to something that you should seek treatment on. Get some intravenous antibiotics. Worst of the worst, then. This is why that little plush microbe had a knife and a fork on it. Necrotizing fasciitis, also called flesh-eating disease. The strains that cause necrotizing fasciitis are very rare, but they have a lot of exoenzymes that work really quickly. So if you can catch this infection early on, it's very treatable. Unfortunately, early on, it doesn't really look like anything that's life-threatening. You have kind of a little redness or a little pain in one area. And then again, those exon enzymes start to be produced, and it's very quickly digesting the tissue around there. Treatment for this, lots of antibiotics and cutting out the infected tissue. So you have to kind of stay ahead of it. Really high mortality rate. Again, thankfully, pretty rare. Okay, and then moving on into more kind of soft tissue infections, strep throat. So streptococcal pharyngitis. Of course, not all sore throats are strep throat. A lot of them are viral. But if you suspect that you may have strep throat, that's something that needs to be treated. Not something you're just gonna, gargle vinegar for or whatever, because there's some complications that we'll talk about here. So usually you're gonna be in close contact with somebody else that had it through droplets. Sometimes you'll see some pus pockets back in the back of the throat. Not always, but some strains will produce this pyogenic. So pyrogenic means fever inducing, pyo means pus inducing. It's kind of nice when you do see that, because then oh yeah, it's strep throat. Yep, we need to go to the doctor. Sometimes, of course, it's a really severe sore throat. Sometimes you get vomiting associated with that, especially in kids, not maybe a little bit less so in adults. Swollen lymph nodes. They will do a strep test on you at Meijer now. So you can go to Meijer, get a strep test, and if it's positive, they'll prescribe antibiotics. So I know for me, I'm pretty prone to this. So I was always like, do I need to go to the doctor? Do I not? Sometimes you wait a little bit longer than you should, but there are secondary infections that are common, bronchitis, pneumonia, and if you've got a strain that's got that erythrogenic toxin, then scarlet fever. Now, luckily today, like I said, there's not a lot of strains that cause scarlet fever. We think those strains were more prevalent like in the 1800s and early 1900s, and they've somehow been replaced with strep that kind of spreads a little bit more easily, but it's not as serious as us. So what does scarlet fever look like? Again, because of that erythrogenic toxin, you get this bright red rash. That's a big scream again, that you need to get treatment for that. Apparently strawberry tongue is another symptom of that. Helen Keller apparently lost her sight and her hearing as a result of having scarlet fever. So this was a potentially deadly infection all through the 1800s, and again, luckily it's not as severe now. That strain was also carried through milk that hadn't been pasteurized. So again, pasteurizing became more common, and 20s, 30s, and 40s really cut down on strains of Strep Pyogenes that could cause this. Okay, so it was usually, again, you have strep throat for a few days and, not treating it, and then all of a sudden this rash will show up. Other complications of strep throat, and again, usually it's either an untreated strep infection or one that's only partially treated. So take antibiotics for a couple days and then don't take them anymore. Most common in children here, rheumatic fever. I was doing a little more reading, and apparently these nodules under the skin, actually they look painful. They're not very painful, and it's very rare for that to happen as part of rheumatic fever. But you do have painful swollen joints, and this is where the antibodies that I was talking about that typically will target the heart valves rather than the kind of cross-react. Antibodies that we make to the Strep Pyogenes will cross-react with heart valves, and it can cause permanent heart valve damage. So it's really, again, something that we really want to avoid. We want to treat our strep throat. And the other complication is acute glomerulonephritis. So these antibodies that we make to target the Strep Pyogenes, for some reason, don't kind of get filtered out the way they should, and they kind of stop up they get all congested in the kidneys and can cause kidney failure. So blood flow obstructed, high blood pressure. I had a friend that every time he got strep throat, he would land in the hospital with kidney failure. So again, some people tend to be more susceptible to this, can be permanent. So we're going to treat this with immune suppressive drugs, immunosuppressive drugs. Okay, we will pick up there after Thanksgiving. Remember, exam on Tuesday. Come see me if you have any questions, and have a good weekend.